Welcome back to Bio 255 Microbiology Laboratory. Today we are going to combine both Session 7 and Session 8 setups, and then we will read their results together as well. Session 7 is entitled Control of Microorganisms. Session 8 is Boiling for Sterilization. But before we begin, let's take a look at our learning objectives. First of all, I want you to gain a lot more experience in culture transfers, aseptic culture transfers, moving bacteria from stock cultures onto new media. Number 2, I want you to be able to assess the efficacy, that means the effectiveness of various chemical and physical antimicrobial agents. These are going to include, in terms of chemical agents, commercially available cleaning products. We are going to take a look at salt as an osmotic stressing agent to control or inhibit microbial growth. We are also going to control growth through nutritional requirements. As far as physical antimicrobial agents go, I want you to be able to assess the effectiveness of boiling as a disinfectant. I want you to take a look at various radiation processes for disinfection. The one we are going to actually implement in lab is ultraviolet radiation of some samples of bacteria, and we will talk about several others. I also expect you to understand the mode of action of these antimicrobial agents. How do they actually cause inhibition of bacterial growth, or actually some of them may actually kill bacteria. So we use two terms, bacteriostatic and bactericidal, to indicate whether they inhibit bacteriostatic processes or whether they kill bactericidal processes. So I expect you to know the mechanism by which they are either bacteriostatic or bactericidal. Number 3, you should be able to explain the difference between disinfection and sterilization. Number 4, I want you to compare and contrast antiseptics versus disinfectants. And finally, number 5, understand how various prokaryotic cell structures, such as slime layers, such as biofilms that develop in bacterial growth cultures, such as spores that occur in certain genera of bacteria, can actually affect the efficacy of one of these chemical or physical antimicrobial agents. Okay, so let's take a look at the bacteria we will be working with in today's lab. The first 2 bacteria we are going to work with again and again and again, and I want you to become so familiar with these 2 that you don't even have to think about their gram stain. I know we have not completed gram staining yet, but basically the bacteria we work with in this class, for the most part, are divided into 2 different gram stain groups. It's based on their cell wall structure, and we will consider that later in lecture and in lab. For now, just note that we have a group of bacteria we work with that are called gram-positive bacteria, and we have a group that we work with called gram-negative bacteria. We are always going to have a control organism for each group, and that control organism is going to be the same. For our gram-positive bacteria, we are going to use as the control Staphylococcus aureus, which you are familiar with from our simple staining procedure we did last time. So Staphylococcus aureus, I want you to know always is gram-positive. Escherichia coli, we also used in our last session where we stained E. coli cells, or in session 5, I believe it was. And I want you to know that that is going to be our stock control for gram-negative organisms. So E. coli gram-negative, Staph aureus gram-positive, EC negative, SA positive. And we will write those on our plates as we proceed in lab this semester. Next, Bacillus subtilis, you actually did work with in your simple stains as well. If you remember, it was a fairly long, thick rod, in other words, a bacillus, that also forms spores. We saw some of those egg-shaped bodies in our bacillus smears when we did our simple staining, both the basic, in the basic stain. We did not stain it in the negative stain, but we did a simple basic stain of crystal violet and one of saffron, and we saw those hollowish egg-shaped bodies that Bacillus subtilis forms. And at the time I mentioned that it forms these when the environment gets very harsh. Either it doesn't have the proper nutrients to grow, or its waste is built and become toxic, or radiation is high, or the environment is very dry. So these can lead to spore formation and dissipation of the vegetative rod form. And what's left are these hollow eggs. Throughout this session and the next, we are going to see just how difficult spores are to kill. They aren't actively metabolizing living vegetative cells. They are in fact dormant suitcases, if you will, of wrapped DNA, the bacterial chromosome, some ribosomes, and then they are wrapped and wrapped and wrapped. And we will discuss that in lecture. However, spores give us fits when it comes to trying to disinfect and sterilize. So they play a really important role in how effective an antimicrobial agent actually is. And we need to consider what we call sporicidal processes and non-sporicidal processes for disinfection and sterilization. Okay, and then finally Serratia marcescens. This is our old friend. You know at room temperature it produces a reddish-orange pigment, and that's why I've chosen to use it in today's lab, so that we can see it if we grow it at room temperature. And there is one instance when we are going to use Serratia marcescens today. It is in fact a gram-negative organism, closely related to Escherichia coli. Okay? So let's go ahead and let's begin setting up all of our cultures. Okay, let's begin session 7 by inoculating our carbon source plates. If you recall in session 6, we actually poured what was called a minimal salt agar plate. This particular plate contains nitrates and ammonium ion for nitrogen sources. It contains phosphates for phosphorus sources. It contains sulfates for sulfur sources. The one thing it didn't contain of our 6 bioelements was carbon. So this is called minimal salts. It does not have a carbon source in it, which we know is the most essential of the bioelements for building the backbones of our macromolecules. So this one is really going to be our minimal salts agar control plate. In fact, I'm going to write control on it, because what I mean by that is we do not expect heterotrophic bacteria to be able to grow on this plate if there's no carbon in the medium, and they cannot fix carbon out of the atmosphere like plants, blue-green algae, and autotrophic bacteria, then we shouldn't see any growth on this. Nonetheless, we're going to divide it. I'm going to divide every plate. Let's see, let's divide every plate in half, and on one half I'm going to write SA and put a plus sign there. That means it is our control gram-positive organism, Staphylococcus aureus. And over here on this side, I'm going to write EC, and I'm going to put a little negative sign on there until you guys get so used to which one's positive, which one's negative. Escherichia coli is gram-negative. Now, positive and negative do not denote good and bad, and I don't want you to think of them like that. It's not a positive result or a negative result. It simply means that Staph aureus has a gram-positive cell wall and Escherichia coli has a gram-negative cell wall. They're chemically different cell walls. That's all. So they may behave differently in different, under different environmental stressors. So what we are going to do is I'm going to inoculate this control plate with Staph aureus and with E. coli. And over here I have my stocks. Now before I began, I disinfected my workbench and I washed my hands. I'm not going to wear gloves today. If I spill, I want to know it. And I will wash my hands before I transfer that bacteria to any other equipment or surface. So I've lit my Bunsen burner, I have my goggles on, I'm going to flame my loop, and I'm going to pick up the stock culture that says Staph aureus or SA on it. Okay, now recall that when you go into a broth culture, you just grab a loopful at the top. You don't stick the loop all the way down to the bottom. I'm going to grab that loopful. I'm going to streak right down the middle of that half of the plate. And voila, that is done, and I can turn it right over, and that liquid will soak into the agar. What do I do next? I flame my loop. Aseptic technique. Flame before and after. Okay, so now I need my E. coli broth culture. Notice how close I am working here. I've got my broths in a bucket right beside my plate so that I can transfer quickly and over a short distance on my lab bench. I go into the broth, grab a loopful, bring it out. I'm going to go ahead and put my stock back in the bucket. Working with broth is tricky. You have to be careful you don't spill. I'm going to take that loopful and I'm going to streak it on the other half of the plate. Turn it over, and that's done. Okay, so my control plate, I do not expect to have any growth, but we have to set that up because who knows? Maybe they can fix carbon out of the atmosphere. We are going to find out. But let's take a look at our other 3 carbon source plates. We have one that was minimal salts agar that we added sucrose to. Minimal salts agar, you will recall, was a defined medium. Once I added an unknown quantity and unknown purity of sucrose, it became a chemically undefined medium. So I'm going to inoculate that. We have a minimal salts plate in which we put Splenda. Splenda is known as sucralose. The molecular formula of Splenda or sucralose contains 3 chlorine atoms. It's actually known as trichlorosucrose. It is very similar in chemical structure to sucrose. But those chlorine atoms are going to potentially make a difference on whether or not bacteria can use Splenda as a carbon source. We're going to find out if that makes a difference. And then finally, we grabbed white vinegar and we placed that in the minimal salts agar. So we actually found 3 carbon sources that you commonly find in your kitchen, and we added them to minimal salts agar, knowing now that there is carbon, the element carbon, available for heterotrophic bacteria. The question is, can they use all of these carbon sources to build the backbones for their macromolecules and hence their cell structures? We'll find out. The carbon's there, but is it actually in an accessible form for their metabolism? So, just as I did on the control plate, I'm going to go ahead and I'm going to divide these plates in half, and I'm going to put SA plus and EC minus on each of these plates. SA is our gram positive, EC is our gram negative, control organisms. SA plus, EC plus. I have this feeling if I say it enough times, you guys will remember it. Okay, so then what do I need to do? I need to inoculate these. And one thing you can do is after you flame your loop, and let's see, I've grabbed EC, Escherichia coli here first. Notice, I'll mix it up a little bit. Notice the turbidity. That is a very, very thick culture of E. coli. So I'm going to be careful, but I am placing E. coli on a sterile medium in a single streak. The question is, do I have to flame my loop again before I go back? And the answer is no. If you do this quickly, there's no reason to do that. I'm just going to grab another loopful, put it on the next plate, and then I'm going to grab one more, Same stock, same loop, onto sterile medium, no need to flame between. I'm working efficiently, which in healthcare we know is extremely important. And then now that I'm done with those three, what am I going to do? I'm going to flame my loop, and I'm going to do the same exact thing for Staph aureus. So I'm going to grab, you have to read your labels very carefully because one of these says SM for Serratia marcescens. This says SA for Staph aureus. And I'm going to go in here and I'm going to do the exact same thing on the other side that says SA. So again, not flaming between. Put it on the vinegar. I could actually even smell the vinegar when I open that plate. Vinegar is acetic acid. White vinegar is made by the fermentation of grain ethanol, grain alcohol, and it is mostly, I think it's 5 to 10% acetic acid. Acetic acid has a 2-carbon backbone, so it definitely provides carbon. But when you've got 5 to 10% acetic acid and we have a chemically undefined medium here, you should be asking yourself, what did Marla do to the pH of this medium? I may have added a carbon source that the bacteria could potentially use, although I just told you it's formed through fermentation, and that's a waste product of bacteria. So we'll see if some can use acetic acid, and even if some can, is this too high of a concentration of acetic acid for bacteria? One of the things we're going to have to consider as far as growth factors that control bacterial growth is pH. We'll look at that later. For now, we've got our carbon source plates inoculated. We are going to place them in the incubator at 37 degrees. We'll do that after we set up our salt plates. I'm going to go ahead and pull out our salt plates now. And remember that what we did when we made our media in session 6 is that we actually created a 5, 10, 15, and 20% solution of sodium chloride with our beef extract, our agar, and our water. And then we poured those plates, we swirled them around and poured the plates, and here they are. I have included here a nutrient agar plate. We made a lot of nutrient agar plates in that session as well. And I told you at the time that in this class, if you do not see a label on a plate, you assume that it is nothing but nutrient agar. And so this does not have any sodium chloride in it. So I'm going to label this 0%, and that is actually going to be our control plate because we actually know that both Escherichia coli and Staphylococcus aureus grow well on the complex medium known as nutrient agar. So this is our control. Our question is, as I added salt to the medium, I increased the hypertonicity of that medium. That medium is becoming increasingly hypertonic as I added enough salt to be 5%, 10, 15, and 20% sodium chloride. You can do this with many solutes. It doesn't have to be salt. In fact, as I say this, I'm thinking about jams and jellies. They are filled with sugar, just filled with sugar. And sugar acts like salt in that it creates an osmotic stress in jams and jellies. And how might osmotic stress affect the growth of bacteria? In fact, the high sugar content is put into jams and jellies on purpose to reduce the ability of bacteria to grow. Question is, how does high solute contributing to hypertonicity affect bacterial cells? Well, we'll find out, and we'll talk about it when we look at these results. What we need to do right now is we need to consider on our plates here what happens to the growth of a gram-positive bacterial organism as we increase hypertonicity, and what happens to a gram-negative one. as we increase. So again, our controls, SA plus, EC minus. So I would just quickly write that on each of these plates, SA plus, EC minus, SA plus, EC minus, SA plus, EC minus. Are you getting the idea? Are you sick of me saying this? SA plus, EC minus. I'm going to drill that into your heads so that you remember. Gram-positive Staph aureus, gram-negative E. coli. So basically, now that I've got the plates labeled, what do? I'm going to turn these over so I can make this very fast. I'm going to line up my plates, and EC is now on all of these. I'm going to grab in the same position, flame my loop. I'm going to grab some liquid here, streak the E. coli side, and without flaming my loop again, I am just going right back into the stock and grabbing another loopful, because I am placing that loopful on a sterile medium. So I'm not worried about contaminating my stock. And this allows me to very quickly and efficiently streak all 5 plates. All right, so I have to flame my loop certainly before I go back into the Staph aureus stock. So once I've done that, I cool it down here, grab my bacteria, and remember clamshell technique? Whoops. Remember the clamshell technique? And basically, I'm just lifting the lid off above it so that nothing can fall into it. But I can just do this too. Remember that? Clamshell, 1 streak to release the loop of Staph aureus, flame it to sterilize it, down on the bench. Okay, these plates can be turned over and must be turned over, remember. They have to be incubated upside down. I'm going to put them in their order from 0 to 20. We now have carbon source plates and salt plates, and they need to go in the incubator at 37 degrees Celsius. Another experiment we are setting up today is the effect of ultraviolet light on bacterial growth. We have 2 nutrient agar plates here. Notice they are unlabeled, so I know they are nutrient agar. We are going to use the organisms Bacillus subtilis, which you already know is a spore former, and Serratia marcescens, which you already know will turn reddish-orange at room temperature. So the first thing I'm going to do is I'm going to label these plates BS for Bacillus subtilis and SM for Serratia marcescens. In your lab book, if you are opened to session 7, you'll see that it asks you to make several lines across the bottom of each plate. And you're going to place a 0 on one of those lines, 10 on the next, 20 on the next, and the 4th line, which is the last line, you write a 30, and down here beneath it, we're going to write 60. What each of these numbers represents is the amount of time we are going to expose that area of the plate to UV radiation. I'm going to do the same thing here for Serratia marcescens. 0, 10, 20, 30, and 60. Now, we need to create something called a lawn of bacteria. A lawn of bacteria is just a thick matted growth across the entire plate where colonies are actually not differentiated. They're not isolated or separated. There are so many bacterial cells we're going to lay down on the surface of this agar that they grow into each other and no clear colonies will be visible. That's a mat or a lawn of bacteria. And to do this, we need to use something called sterile swabs. You guys know these in healthcare. They use them all the time. So right now they're sterile and they're wrapped. I have a broth culture here of Serratia marcescens. And you can see it's labeled SM. So I'm just going to hand vortex that to mix it up. And I am going to open up my sterile swabs. They come 2 in a pack. So I'm going to open them at the handle end, not the swab end. I'm going to remove one of these swabs, and I'm going to very quickly put it into the broth culture of Serratia marcescens. Now, the thing about broth, remember, is there are trillions of cells in every tiny drop. So I'm going to hit it against the side to make sure I don't have any drops that will fall on the bench or on me. I'm going to place that broth upright back in our stock tub, and I don't need this gas, so I'll turn that off. I am going to take my Serratia marcescens plate. I'm going to swab it back and forth from side to side. I'm actually touching the sides of the plate, and I'm going to do that back and forth, and I'm going to roll the swab around as I do this so that I am sure to make a thick layer of bacterial cells on there. Then, whenever you make a lawn, you turn the plate 90 degrees and you do it all again. I don't need to dip back into the broth. I am laying down many more cells as I do this. Okay, do it a couple of times, and then when you think you're done, you're not. Last thing you're going to do, turn that plate 45 degrees and do it again, just to ensure you've got complete coverage. Okay. All right. So that's Serratia marcescens. I'm going to move that out of the way. Question is, what do I do with this? I have to ask you guys something. In healthcare, do you ever re-sleeve anything? Needle swabs, do you ever re-sleeve? No. So this needs to go in the disposal. Let's walk over to the disposal at the side of the room, because I want you guys to see where our microbiological waste is located. And I know I've shown you the placement of contaminated tubes for broth and agar, but right here where it says microbiology waste, this tub is where we put our plastic petri dishes when we're done with those cultures, and where we put things that are not made of glass, such as this swab. Okay? So that goes right in there. From my hand to the bin. I'm going to go ahead and I'm going to bring the bin over to our lab bench, so when I swab the next one, I can simply put that directly into the waste. But I wanted you to see where it's located in the lab. Okay. So let's take a look at our last plate here, which is Bacillus subtilis. Bacillus subtilis is, we said, our spore former. It is also a strict aerobe. And so if you look at this broth culture, it may not look turbid. It may look kind of clear, except for a thick mat of growth at the bottom. That actually was sitting at the top before I moved the stalks today, and it fell to the bottom. Bacillus subtilis is a strict aerobe, so it only grows on the surface of the broth where there's oxygen. And when you jiggle it, that falls to the bottom. I'm actually going to mix it up so I can ensure that when I grab the swab full, I'm getting loads of those cells. I'm going to take the remaining sterile swab out, reach in, get it nice and moistened, tap it on the side so I don't drop any liquid, place that back in the stalks, and then what do I do again? Recall. you're going to go back and forth on top of your previous streaks, and you're going to do this several times while you're swirling the swab around back and forth on all sides of the swab. Then you're going to turn the plate 90 degrees, do it all again several times, covering 100% of the plate. And then what do? 45 degrees, and you do it one more time. Okay, so now we have our 2 lawns of our bacteria that we are going to test against ultraviolet light. I'm going to take my swab and I'm going to toss it into my disposal bin, okay? Let's meet over at the ultraviolet radiation box in the back of the room, and I'll show you how we are going to ensure that each of these zones has UV light applied to it for the appropriate amount of seconds. 0, 10, 20, 30, and 60 seconds, 1 minute. Okay, here is our ultraviolet light box. You definitely don't want ultraviolet light in your eyes, so we have a glass here that's going to block that, and so we will not be turning on the UV bulb until everything is closed up. But right now, to get started, we need to take our plates, our Bacillus subtilis and Serratia marcescens lawns, and we need to get them under the UV bulb. I'm going to open the box up like this, okay? Lift this up so that I have good access to the ground here, the countertop. Now, I cannot get UV light to penetrate plexiglass. So first of all, I'm going to have to turn these over and I'm going to have to remove their lids. So we'll put those aside. This is Serratia marcescens, 0 seconds is at the top. Bacillus subtilis, 0 seconds at the top by the label. So I'm going to back them up against each other, 0 to 0. And the first thing that I'm going to do is I'm going to expose only the bottom, which was the 60-second area of the lawn. So I'm going to use that note card to cover everything except the spot that said 60 seconds. And now we need to back up and close the glass shield, close the doors. And here I have my cell phone. I am going to watch that for 10 seconds. I'm sorry, for 30 seconds. We're going to let it go for 30 seconds. I'm going to flip this on and start when the light turns purple, and you see it turn purple. I'm going to start the watch. We're going to go 30 seconds, not 60, and you'll see why in a moment. So here we go. When the light comes on, we hit start. We'll let the UV hit the exposed bacterial lawns for 30 seconds. Ultraviolet light causes the formation of something called thymine dimers in DNA strands. And those thymine dimers form only where 2 thymine bases are side by side or adjacent along the strand. So if the sequence of bases includes thymine, then a dimer may form. That is going to cause a bubble in the strand, and the bacteria are going to have to repair that if they are going to avoid mutation. All right, so we did our 30 seconds, actually a little over. I see I went 34 seconds because I was talking. I'm going to spread the plates apart, and now I've exposed the 60-second and the 30-second region. At this point, we are going to just do 10 seconds of exposure, and that'll make it 40-second exposure to the 60-second realm and 10-second to the 30. Not what we want, but we will get there. Watch, I'm going to turn on the light, start the timer. I'm going to watch the timer. When 10 seconds is up, I'm going to shut off the light. So I'm watching the timer, 8, 9, 10. Okay, we open this back up. We spread our plates apart. Another section exposing the 60, the 30, and the 20. And now, whoops, I close this, reset my timer, and we are going to do this for another 10 seconds. So here we go. Now, at the end of this period, the 60-second region will have been exposed for a total so far of 50. Okay, turn the light off. Whoops. Open it up again. Let's think before I start this up again. I'm going to move it out to the 10-second realm. And let's think about this. At this point, this region, the 60 seconds, has been exposed for a total of 30 seconds, 10 seconds, and 10 seconds. That's 50 seconds. It needs one more 10-second exposure to be a total of 60 seconds or 1 minute. The 30-second realm so far has only been exposed for 20 seconds, and the 20-second has been exposed only for 10 seconds. So right now, what I've got here is the 10-second strip is opened up and ready to be exposed for 10 seconds. When I'm done here, the plates will be finished. 10, 20, 30, and 60-second exposures. The zeros remain covered entirely. So we're going to shut this back up. I'm going to reset my stopwatch. I'm going to turn this on when it turns purple. Start it up. There we go. And 5 seconds, 6, 7, 8, 9, 10. There we go. We are done. What we have to do now is be very careful not to touch the surface of the plates. We're going to remove the card. I'm going to place the lids back on. Immediately turn them over. Now, I want to be able to see serratia well, and if you recall, it was pretty flat, transparent colony morphology when it grew at body temperature. So I'm going to leave this out at room temperature so it turns orange. So we'll leave this out at room temp and this out at room temp, and we'll come back in a couple of days and we'll look at the level of their growth based on the time of exposure to UV light. Okay, for this section of session 7, we are going to test various antimicrobial agents, very common antimicrobial agents, and you were asked to bring a cleaning or disinfecting product from your household to today's lab. Because we cannot canvass the entire class's household cleaning products, instead of doing 24 different cleaning products, we are just going to do the 4 I have in front of us. So I've brought some hydrogen peroxide. You guys probably have this in your medicine cabinet at home. Hand sanitizer, really big on hand sanitizer these days for obvious reasons. So I've got this hand sanitizer, not a product we're familiar with. We've been purchasing things in bulk in America now. A lot of it is made out of country. I don't know this brand, but it does have 70% ethanol, and anything 62% ethanol or up to 70 is pretty common in hand sanitizer. So ethyl alcohol, 70% right here, among other aloes and so forth to moisturize hands. Here we have your 10% bleach bottle from your drawer, and finally we have Bactine, which you can find in the drugstore. And so I want to talk about the difference between a disinfectant and an antiseptic, but before I do so, I need to talk about what disinfection is. Disinfection is the removal of the majority of pathogens to a level at which the risk of infection is quite low. So it is either the majority of the pathogens, sometimes it's all microbial pathogens are removed through the process called disinfection. And that's very different than sterilization. Sterilization means the complete removal of all life forms, viruses, prions, fungi, bacteria, archaea, everything's gone in sterilization. There's no living component at all if you have sterilized something. But for the most part, we don't sterilize things in our lives. we do want to disinfect them. We want to remove the majority of pathogens so that the risk of infection is very, very low. Most often we want it to be absent if we can. That means we're removing pathogenic microorganisms, not, and those are usually bacteria and viruses, not necessarily all microorganisms. In fact, we know many disinfectants and antiseptics remove pathogens with the exception of bacterial spores, and some spore formers are pathogenic. So when we look at the 4 products we have out in front of us, their purpose is disinfection. The question is, are they a disinfectant or are they an antiseptic? So here I want to clarify the difference. A disinfectant causes disinfection on inanimate objects. Inanimate objects are non-living material. So this workbench is inanimate. This test tube rack is inanimate. My reading glasses are inanimate. Faucet handles, door handles, shower stall floors, etc. Those are inanimate objects that we commonly want to disinfect. An antiseptic, on the other hand, disinfects living tissue. So antiseptics you can apply to living tissues. So right off the bat, I look at this and I think, well, hydrogen peroxide I can apply to living tissues. We can pour this in wounds, can't we? It comes in a brown bottle at the pharmacy. It contains 3% H2O2. That's the chemical formula for hydrogen peroxide. And it bubbles away when you put it in cuts and scrapes. Some people even gargle with this. So we can consider this an antiseptic, but can you use a hydrogen peroxide or peroxide as a disinfectant on an inanimate object? Yes, you can. So this compound doubles as both an antiseptic and a disinfectant. When we look at this hand sanitizer, we're not going to be cleaning the surface of the desk with ethanol. This is 70% ethyl alcohol. It is used on human tissues, on the hands. Isopropyl alcohol is another example you use in healthcare. Those are those little alcohol swabs before a needle goes in, 70% isopropanol. So those are considered antiseptics. There are times, though, when alcohols can be disinfectant. When I was a little girl, you got your temperature taken at the pediatrician's with a rectal thermometer. That went in one end after another of many different children. In between taking their temperature, what did they do? They stuck it in a bottle of isopropanol through cotton, and they removed it, wiped it off with isopropanol, and stuck it in the next child to take their temperature. We don't do that anymore, thankfully. But that's an example of alcohol being a disinfectant, isn't it? That thermometer was an inanimate object. So again, we could consider alcohol in general, like hydrogen peroxide, both an antiseptic at times and a disinfectant at times. But this bottle of hand sanitizer is meant solely as an antiseptic. 10% bleach. 10% bleach is solely a disinfectant. This is something you should not put on your skin. Why? Sodium hypochlorite is corrosive. It'll take off the top layers of your skin. We use it to disinfect laundry. We use it on inanimate objects. You and I use it in this class on the surfaces of our lab stations. So this is what I told you in the very first day, a very effective disinfectant. And finally, we had Bactine. Bactine is a product, and by the way, this is not a product endorsement commercial, so don't think I got paid. They're not sponsoring me, but Bactine is an antiseptic. This is specifically so that you can put this on scrapes and wounds and so forth to disinfect the wound. So because it doesn't harm the living tissue, it is an antiseptic. It's not of use to us on inanimate objects. So let's go through this. We've got an antiseptic for wounds. We've got a disinfectant that we use all the time in healthcare, in the microbiology laboratory, at home, in your kitchen sink, in your toilet, and so forth. 10% bleach is wonderful. That is a product endorsement. Sodium hypochlorite. Hand sanitizer, this is an antiseptic, although we know alcohol in general can be used as both an antiseptic and a disinfectant. And we've got hydrogen peroxide, which also can be either an antiseptic if you pour it in a wound, or if you're using it to get blood out of a fabric or whatever, it's being used as a disinfectant. Okay, so those are the 4 disinfection chemical agents we are going to use today. I'm going to move those out of the way for a moment, and I want you to take a look at the setup I have in these 2 test tube racks. We're going to work with 2 bacteria, Staphylococcus aureus, our control gram-positive. We're going to use another gram-positive organism. So we are comparing gram-positives with gram-positives. The other one is Bacillus subtilis. It's also gram-positive, but you know that Bacillus subtilis is a spore former. In fact, the genus Bacillus, all member species of the genus Bacillus are spore formers. So we're going to compare a sporulating gram-positive organism with a non-sporulating gram-positive organism. And I have in front 4 empty sterile test tubes. They've been autoclaved and they're sterile. So I am going to take in each rack here, I'm going to take some hydrogen peroxide, and this is not particularly controlled, but I am going to pour into this tube about a thumb's width worth of hydrogen peroxide. And I am going to label this SaH2O2. Now, I'm going to do the same thing over here with the tube that says BS. I'm going to put H2O2 in here. And I will again pour a thumbnail's worth of H2O2 in there. Okay? All right. So that's set up. Next, I'm going to take our hand sanitizer. I'm going to squirt hand sanitizer. It's a gel, so it's a bit of a problem getting that down. I don't want to the bottom. I don't want to use too much of that because it is a gel. And I'm going to put hand sanitizer, or HS, under that. Under a BS here, I'll put HS again for hand sanitizer, and I will squirt that in. We'll let that settle to the bottom. We'll move that out of the way. We've got bleach. In our 3rd empty test tubes under SA, I'm going to write bleach. I'm going to squirt thumbnails worth of bleach in there. Again, not completely controlled, is it? I ought to be ashamed. Shockingly, I'm not. That's close enough for government work. And there we go. Now, the Bactine. The Bactine, get the childproof lid off, squirt that in, and write Bactine under SA. And in the red rack under BS, we write Bactine and squirt that in. Okay. So we are ready. We've got our 4 disinfectants or antiseptics. Recall our stock cultures. Let's see. I have Bacillus subtilis here. I'm going to put that stock right there. And I have Staph aureus right there. Now, let me tell you how this is going to play out. So I am going to inoculate each of these 4 tubes, each of these 4 disinfection chemicals with Staph aureus. I'll probably put 2 loopfuls in each of these, okay? I'm going to put 2 loopfuls of Bacillus subtilis over here in these 4. Same 4 disinfectants or antiseptics as over here, using this stock. But I'm not going to do this till I am ready to rock and roll because I have to time this procedure. So I actually do have my cell phone here. I will disinfect it after I am done just to be safe. But this is actually my stopwatch, okay? Now, after I inoculate these, and I'm going to inoculate these in sequence, I'm going to do the Staph aureus set first. I'm going to inoculate 2 loopfuls here, 2 loopfuls here, 2 loopfuls here, 2 loopfuls here. I'm going to start my timer at 1 minute after I've inoculated all 4 sets. And when 1 minute is up, I am going to transfer a loopful from here, from our original disinfectant tube, to this tube of nutrient broth. Remember, you made nutrient broth when we prepared media. You made nutrient broth tubes and we put them in the autoclave. These are those tubes. And this is why we needed them. And I've already labeled this SA for Staph aureus, H2O2 for hydrogen peroxide, and 1 for 1 minute exposure to hydrogen peroxide. And so I'll transfer all of those to each of these tubes. This tube, SA hand sanitizer, HS, 1. This tube, SA bleach, 1 minute exposure, and of course SA Bactine, 1 minute exposure. And as soon as I do that, as soon as I've inoculated all 4 of these, I'm going to keep the timer running because these are still sitting in their tubes. But I'm going to take these and I'm going to move them over to this rack once I've inoculated them so that I can put them in the incubator later. But I'm just going to sit them there and keep that going. But these tubes, the bacteria are still being exposed, aren't they, from the initial inoculum? So when the stopwatch gets to 5 minutes, I'm going to take another loopful of each of these and transfer it to this set of 4 tubes. And guess what they say? SA H2O2 5, meaning this tube represents the bacteria that were exposed to the disinfectant or antiseptic for 5 minutes. It's now in nutrient broth, no longer exposed to the disinfectant. I'm going to move it over here. When we're all done, we will put these in the incubator. I'm going to keep my stopwatch going. When 10 minutes is up, I'm going to do the same thing into these tubes that are marked SAH2O210 and so forth. And when 15 minutes is up, in the back tubes. So let's get started. And I'm going to go ahead and start up my fire. I've got my goggles on. I've got my loop. And I've got my stock of Staph aureus. So I'm going to just hand vortex that, mix it up, flame my loop, get it nice and clean. And here's what I do need to make sure I do. Okay, I'm going to go ahead and add a couple of loopfuls of this to the hydrogen peroxide. Tap it. I need to flame between because I want to get rid of the chemical. Since I'm putting 2 loopfuls in initially, okay, and flame that. Before I go into the hand sanitizer, I had to flame it. I don't want hydrogen peroxide on that loop. We are testing the efficacy of each of these separately. I'm going to flame it here because I don't want to put hand sanitizer back in my stock. So just this once, I know I taught you to hold the stock, but just this once so that I can show it for the video. I'm not going to. Okay, flame it again. Now I've got the bleach. Go into my stock. Grab a loopful. Whoa. Since I touched the edge of the top of that, I'm going to go ahead and run it through the flame to sterilize it. And second, shaky hands. Second loopful, flame it, put it down, put the lid on, grab my Bactine and 2 loopfuls here, flame it so I don't screw up my stock, go back into the stock. Get my next loopful. Okay, flame it again. I'm done. Put the lid back on that. I'm going to start my 1 minute. I realize it is a little bit more than a minute from when I started inoculating it, but we're all going to do these the same, and we're assuming that the amount of time it takes me to do the transfer is roughly equal each time. Okay, so we're going to wait. And we're going to watch, and in 1 minute's time, I'm going to have my loop ready, sterile, ready to go. And in 1 minute, I'm going to move a loopful of this tube into this tube. So I'm just going to have it ready to go. So remember, I'm moving from the hydrogen peroxide into the nutrient broth. And here we go, we're at 60 seconds. So I'm going to grab a loopful there and transfer it. This tube is done. It goes over here. Flame my loop, grab the next set. Transfer from the hand sanitizer over to the nutrient broth. Put the lid back on, transfer that over, flame my loop. While I'm flaming that, I'm going to grab the next one, next set. Go ahead and transfer from the bleach over. One loopful, flame my loop, get that tube set aside, grab the Bactine. Did you guys see how this is going? Okay. And now we're waiting. I'm watching the clock. And as I watch this clock, it's already at 2 minutes. When it gets to 5, what do I have to do? I have to start the next round of transfers. So here are the 1 minute. And put this back. And at 5 minutes, I'm going to do the exact same thing. Okay? Okay, we're back. 5 minute mark is coming up right now. So I'm going to go ahead into the hydrogen peroxide, remove a loopful, place it in the Staph aureus hydrogen peroxide 5 minute tube, flame my loop before I go on, put this aside. Here, we're going to grab the hand sanitizer, do the same thing, transfer quickly, 1 loopful to the 5 minute, put this aside, flame my loop, grab the bleach and the 5 minute tube, and transfer loop. Whoa. There we go. Flame my loop. And grab the Bactine. And I'll see you back at 10 minutes for the next set. Videos are wonderful, so you don't have to wait around. I'll see you in 5 more minutes. Okay, we're back, and here we go again. We've got SA hydrogen peroxide, and we've got SA hydrogen peroxide, the 10-minute tube. I flame my loop. I go into the hydrogen peroxide where now the bacterial cells have been exposed for 10 minutes, and I transfer a loopful to the 10-minute tube, put my caps back on, flame my loop, put this aside, and grab the hand sanitizer along with the hand sanitizer 10-minute tube. And you know the routine now. So I'm going to go in, cool it down, grab a loopful of hand sanitizer, transfer that over. There we go. Lid's back on. Put this aside and flame my loop. Grab the bleach. These Staph aureus have now sat in that bleach for 10 minutes. Let's see if they are still alive. That's ultimately our goal here. As we move them out of the bleach and into good complex medium, if they survived the bleach exposure, they will grow in that medium. And that is our goal, to see if that is the case. Okay, and finally the Bactine. Loop is flamed. Grab a loopful of Bactine. Transfer. Flame. We're good to go. I'll see you back in 5 more minutes for the 15-minute sample. Then we're done with Staph aureus. I'm not going to make you sit through the Bacillus subtilis because I think you have the idea, but I myself will do these transfers and then we'll read all of the results. when we come back. Okay, it's almost 15 minutes, so I'm going to go ahead and flame my loop. And I'm going to go into, right now, I'm going to go into the hydrogen peroxide, transfer a loopful, our final loopful, into nutrient broth, and flame my loop. Get that over here, and grab the hand sanitizer. Do the same thing here. Recall now that Staph aureus has sat in that hand sanitizer for a total of 15 minutes. We'll see if that was enough to kill it. Put that back, grab the bleach, and transfer the bleach. Quick transfer. Flame the loop, put that aside, put that back, and finally our Bactine. Grab one loopful, transfer, and we are done with that set. Now, I'm not going to make you, as I mentioned, sit through the rest of this with the Bacillus subtilis. However, I do want to mention, I'm going to put this out of the way, and I do want to mention here that, that we've got the exact same antiseptics and disinfectants, the exact same setup. I've labeled everything. I will go through this. The difference is Bacillus subtilis is a spore former, remember. So we're going to compare this gram-positive spore former with this gram-positive non-spore former. I will put this rack labeled Staphylococcus aureus in the incubator at 37 degrees, and when we come back, we'll see in what tubes, if any, we have growth. And that will tell us what amount of time we needed to expose Staph aureus to our antiseptic or disinfectant in order to actually kill it. And how effective then was our disinfectant or antiseptic at killing Staph aureus. And we'll do the exact same thing for Bacillus subtilis. One other thing that we do need to do is we need to inoculate a control. So I'm going to grab a tube of nutrient broth. I'm going to label it Staph aureus, and before you leave, right now, I am going to take Staph aureus from my stock here, and I'm going to inoculate the control. Why am I inoculating the control? Because I want to make sure that the bacteria in the stock are viable and that they do, in fact, grow well in this nutrient broth that we made in the last session. Okay? So we will compare all of our other tubes to that control, and I'll put that at the back of the rack. All right, I'll go ahead and finish up the Bacillus subtilis portion of the experiment, and we'll compare both of those when you return. Welcome back to session 8. Today we are going to set up the boiling for sterilization experiment. Before we do so, I want you to think about what we did when we prepared microbiological media in session 6. When we were done mixing all of the ingredients, we dispensed them into flasks, and we put those flasks in something called an autoclave. And an autoclave, we said at the time, was simply a pressure cooker that allowed us to achieve 121 degrees Celsius. And we maintained that temperature for 15 minutes in order to do something called sterilizing the media. What we were actually doing was killing bacterial spores. 121 degrees Celsius is extremely hot, and boiling water at sea level is only 100 degrees Celsius, 21 degrees cooler than the autoclave. So the question we have today that we need to address is, does boiling actually sterilize? Can you increase the length of time you boil something at 100 degrees? And thus sterilize and kill bacterial spores? Or is it more likely just a disinfectant? Or can it be both, depending on the length of time you boil something? I'm sure you all have relied upon boiling at home to sterilize things like dinner plates or dirty laundry, or maybe baby bottles and the nipples that go with baby bottles, we like to think that if we boil those, we are actually sterilizing them, but are we, in fact? We'll find out. So today, what we are going to do is we're going to expose 3 different bacteria to boiling temperatures for various lengths of time. These 3 bacteria you are already familiar with. The first one, Staphylococcus aureus, is our control gram-positive bacterium. The second one, Escherichia coli, you know is our control gram-negative. And the third one, Bacillus subtilis, is also gram-positive like Staph aureus, but what it does that Staph doesn't is it produces bacterial endospores, those dormant bodies. So let's take a look first at our heating block. This heating block has the ability to achieve boiling temperatures, or 100 degrees Celsius at sea level. It is filled with aluminum beads, low heat capacity. They heat up very quickly, and I have placed 9 test tubes of the nutrient broth you made in the previous sessions in there. And although it's not quite at boiling, it is very close. It's around 98, 99 degrees. And so we probably won't get it to boiling in this heat block necessarily, but we are certainly getting it way above pasteurization temperatures that range from about 65 to 77 degrees Celsius. So we're way above that right now, and you can see the steam building in the test tubes. The first 3 test tubes I've labeled with the organism I'm going to place in them, Staphylococcus aureus, SA. And they are labeled 1, 5, and 15. And those numbers represent the amount of time I am going to leave these tubes at that temperature once they have been inoculated. The second set here in the middle, they are labeled in the same way, 1, 5, and 15, but with the organism EC, or Escherichia coli. And finally, over here, we have Bacillus subtilis, 1, 5, and 15. So this media has been sitting for quite some time in this heat block. It's about 98, 99 degrees Celsius. Over here on the right, I have a tub of ice water, and it's filled with ice packs. These ice packs have been sitting here, and they brought this temperature to about 4 degrees Celsius or so, probably lower, I hope. And I've placed a test tube rack in there. So what we are going to do now is I am going to take these stock cultures. I'm going to inoculate the 3 bacteria in their 3 respective tubes. We're going to start the timer on my cell phone, and at 1 minute, I will remove the 1-minute tubes and place them in the ice bath so that they immediately cool down and are no longer at a boiling temperature, so we can guarantee that they've only been in there 1 minute. And we'll do the same 4 minutes later with the 5-minute tubes, and 10 minutes after that with the 15-minute tubes. Okay, and I'm going to cut out and come back after those times arrive so that you don't have to sit here and watch a boiling tube boil. Okay, so first things, goggles on. And got my goggles on, start my flame. I am also going to first, right off the bat, take my Staphylococcus aureus stock tube, and I have here a tube of nutrient broth labeled SA0. This means it's never been boiled. And I'm actually going to use that as my control. So I want to make sure my stock grows in this medium, but I want to use a tube that has not been brought up to any boiling temperature. So I will inoculate that tube, which right now is at room temperature, but I'm going to place it on our ice bath as 0. And I'm going to do that for each of these as we go. So next thing I'm going to do is take a loopful of this stock. Going to inoculate, that's a very hot tube. Going to inoculate the first tube. I do not need to reflame my loop. I can go right back into my stock. Hot tube. Inoculate the 5 minutes. And yikes, very hot tube. The 15-minute tube. Okay. I'm going to flame that. Put my stock out of the way. And I'm actually going to start my timer right now. And while I'm doing that, I'm going to go ahead and inoculate control tubes and the experimental tubes for E. coli as we go. So I get the 1 minute down. Five-minute. Watching my timer because I'm going to have to remove here in a second the 1 minute Staph aureus tube. Okay, so now those E. coli tubes have been inoculated. Flame my loop. I'll wait to do the Bacillus subtilis here because in a matter of seconds I'm going to have to take the Staph aureus out. Okay, and put that in the cold. I was about a half of a minute in when that first tube of E. coli was inoculated. Now I'm doing a Bacillus subtilis, and I'm going to look at that clock at 20 seconds and inoculate that. And go ahead and get the rest of these Bacillus while we're waiting. And it looks like I'm ready to remove the E. coli. And in another, let's see, few seconds here, I'll remove. Let's see, that's the 15. Let's see, did I do the 5? I have to do the 5. Skip that. So let's go back and do that. You can imagine it gets fairly crazy here. And I'm going to go ahead and remove our 1 minute, not quite yet, 1 minute Bacillus right now. Okay? Now, I'm letting this go. I'm going to cut back at 5 minutes and remove the Staph aureus. And I will also inoculate our control Bacillus subtilis and E. coli. Okay, so we're coming up on 5 minutes, and at 5 minutes I'm going to remove the Staph aureus 5 tube and place that on ice. I have gone ahead and placed all of the controls, the SA0, the EC0, and the BS0 in the ice bath. After inoculating those, we have all the 1 minute tubes here. Now we have the 5 minute SA. So we'll wait a few more seconds before we take the EC out because I did those after the Staph aureus. And then we'll give the Bacillus subtilis a few more seconds as well. So just kind of assume we were half a minute or so apart. I'll take the 5 minute E. coli out and we'll let this go a half minute more or so before we take the Bacillus subtilis out. Remember, this entire heating block is extremely hot. Recall that aluminum has a very low heat capacity, which means it heats up very rapidly, and it cools off very rapidly too. But those beads right now will burn you if you touch them. Okay, so I'm going to go ahead and take the Bacillus out as well. And now what's left are the 15-minute tubes. So we're going to cut the video and come back when 15 minutes is up, and we'll remove those test tubes at that time. We're back, and it's almost time to remove the final tubes, the 15-minute tubes. We'll place the Staph aureus in the water bath. We'll give it a few more seconds before we remove the E. coli, and then finally the Bacillus subtilis. When we're all done and we've cooled them all down, it's time to place them in the incubator. And let's recall what we're trying to determine. We wanted to cool them immediately so that they didn't remain boiling. We wanted to see how effective 1 minute was of boiling to kill E. coli, Staph aureus, and Bacillus, 5 minutes of boiling, and 15 minutes. So now what we need to do is we need to place these nutrient broth tubes in the incubator to see if there's anything living in them. If any Staph aureus cells survived, if any E. coli cells survived, and if any Bacillus subtilis cells survived, they will grow up overnight in the incubator in the nutrient broth, and we will see turbidity in the morning. Okay, I'll see you then. So here we are back at the incubator. Let's just wrap up sessions 7 and 8, the setup. We've got our temperature set just about to 37 degrees body temperature, so we're good to go. I've opened the door recently, so that's probably it's down a little bit. I'm going to open the door now, and I'm going to show you our setup. I just placed our boiling for sterilization tubes, the 0 through the 15 minutes of boiling in this rack on this shelf, on the bottom shelf. In the middle shelf, we have our disinfectant antiseptic labs where we expose Staphylococcus aureus and Bacillus subtilis to 4 different disinfectants or antiseptics for 1 minute, 5 minutes, 10 minutes, and 15 minutes. And up here, we have our carbon source plates, minimal salt agar plus our carbon source, and we have our salt plates. And so I'm going to close this door. We're going to incubate those all at 37 degrees Celsius. I have our ultraviolet light plates, Serratia marcescens and Bacillus subtilis, and I'm going to place those on the countertop at room temperature. When we come back, we will read all of these results and talk about these different methods of microbial control.